it's not straight from my drawing or something like that.
They're going to come in and show us how to do a little.
I know how it works. I have the instructions. I'll show you first.
Okay, thanks.
All right, if I could ask everyone to take their seats. I know we'll be joined by many more. This is really an incredible pleasure and honor for me. I don't know how all these gentlemen and gentlewomen got up here, but we are so incredibly privileged to have this remarkable, quite unique group of scientists talk to us about their vision for how developmental adversity can be and should be considered a biological variable. And I'm just going to name names and then turn it over to Dr. Ressler. This is a session, a two-part session, so I hope you'll plan on the whole rest of the morning, chaired by Susan Miller, who is really one of the architects with Dr. Kerry Ressler of this remarkable initiative. And we are joined by Dr. Ronald Kessler, Dr. Bruce McEwen, Dr. Martin Teicher, Dr. Charles Nemeroff, and Dr. Elisabeth Binder couldn't be here, but Dr. Ressler will be providing some of her cogent ideas. So without further ado, and with great honor, I'll turn this over to Dr. Kerry Ressler.
Thanks so much, Julian, for your support, and Michael and Teresa for just a fantastic meeting, and we really appreciate the support for this important work. Developmental adversity is a biological variable. What we're struggling with is, and what we hope to present to you in the next couple hours, is the enormous data set that shows that developmental childhood adversity changes your epigenome, changes your brain, changes your physical, your biology in the body, changes your biology in neurons, and changes the way we respond to treatment. And yet despite this, as a field, we've not yet been able to break through the barrier of having this as a standard approach to basically all science, certainly neuroscience and psychiatry, but really all of medicine. And so our job today is to really think about this problem. We're going to start with Susan Miller, who I'll tell you about in just a second. Then unfortunately, Elisabeth, we made great heroic efforts to have Elisabeth be here. When we found out a couple weeks ago she couldn't make it, Susan actually took a video, but in technical reasons prevented us from showing the video. So I'm going to present the epigenetic work of her lab and our lab over the last couple decades related to this work. Then Ron Kessler is going to talk about epidemiology of individual and populations and how childhood adversity is a critical variable. Bruce McEwen will talk about developmental stress and allostasis. We'll have the group break at that point. We'll regroup. Marty Teicher will then pick us up with brain architecture and sensitive periods, and Charlie Nemeroff on the importance of childhood adversity as a biological variable. And what I think I'm most excited about is then we'll have about 20 minutes for a panel discussion at the end to really think about how do we move forward as a field. So Susan Miller is a remarkable person. After 20 years in international business management, from banking and commodity firms to a wide range of business functions, project finance, and really international work across Europe and America, she followed her true passion in mental health and became a psychiatric nurse at Yale in 2003. And then in 2006 began an organization called ANS Research as a platform for speaking about the devastating impact of developmental adversity on chronic and occurring illness, with Bessel van der Kolk as the first CSO of her program. So with that, I will introduce Susan to get us started and to really have us think through what today's about. Thanks so much.
Thank you, Kerry, and thank you all for being here. My task is to explain why we're doing this session and what we hope to accomplish with it. Kerry has already introduced my background. The first part was business, the second was as a clinician, a nurse practitioner. And the third part is the part that you're seeing now, which was where I and others wanted to see if we could figure out the strategic impediments to bringing the impact of developmental adversity into mainstream science. into our understanding of brains, bodies, illness, and behavior. For disclosure purposes, no one pays me to do this. I want to take just a minute to thank the people who made it possible for this to happen. I don't see Bessel van der Kolk in the room, but if he had not come into my life, we wouldn't be here. Marty Teicher also, and Kerry in particular, has made this wider conversation possible. I think it's an essential step in bringing the biological impact of developmental adversity into our mainstream understanding of science. And we thank Julian Ford for his suggestion that we begin this discussion here at ISTSS today. Thank our speakers and all of you for generously being here. So as the people in the room know, I'm going to just go past my disclosure thing. Okay, as people in the room know, once you've seen the impact of developmental adversity on brains, bodies, and behavior, it's hard to believe that it isn't already mainstream. Being meaning-making creatures, we all have reasons why we think this might be so. I started with the idea that it's a very complex challenge, the integration of this material, and that it doesn't fit into any one silo. As the slide shows here, the complexity of all of this might be best understood by looking at the alterations that are associated with it, just an overview. The brain, almost everything about the way the brain does its work is impacted by a history of developmental adversity. Almost every system of the body, I think it's every system of the body, but I can't be positive, is impacted by this in molecular and cellular ways. It affects health outcomes and it affects life outcomes. The health outcomes, importantly, aren't just in mental health. It's in what we think of as medical health as well. And some of the signature characteristics of a childhood history of adversity is comorbidity, chronicity, complexity, and treatment resistance. When we're talking about health outcomes, those aren't ones that we talk about all the time, but they're ones we think are important. So what we believe, and the people here will help make the case for it, is that the alterations associated with developmental adversity, they're broad enough and powerful enough to confound research results, to make it hard to see the factors that are part of, that are drivers of health and well-being. It's so big, it's so powerful, it masks them. We can't see them until we start to control for developmental adversity and be able to parse that out. It also, importantly, we think, undermines rigor, transparency, and reproducibility. Given the differences in brains, bodies, behavior, etc., it's no surprise that if you're not aware of the percentage of your research population that has this history, it's hard to replicate your results unless you, by luck, happen to come up with the same proportion of people with and without a childhood history of. This is at the heart, I think, of why it's been difficult to change, to bring developmental adversity into mainstream science. And we were late in realizing this, that NIMH helped us to see it. Just as sex and race required a congressional mandate in order for them to be considered as biological variables, so will developmental adversity. It's hard to imagine how this can happen unless somebody decides to take the task on. The people that we thought would be most likely to do it, it turns out, the agencies that know the most about this, the organizations that know the most about this, cannot by law attempt to influence policy. They can educate, but not influence policy. It has to come through Congress in a totally different pathway. It was the same for sex and race. So what Kerry and I decided to do with others, including the people here, our speakers, is to see if we can't figure out what it would take to build a consensus that this was an appropriate next step, to consider developmental adversity as a biological variable in order to go to Congress to ask for this kind of a mandate. And if now's not the time to learn what it is we need to do or to study in order to bring that day forward. So our question for you as an audience, I can't overstate the importance of your response to this. As you hear the highlights that our panel is going to communicate with you, does it make sense to consider developmental adversity as though it were a biological variable? Could doing that clarify the role of adversity in altering brains, bodies, behaviors, and treatment outcomes, thereby reducing research confounds? And could doing this improve rigor, transparency, and replicability? And we really want your responses to this. This is a double session. Our speakers are going to speak each for 20 minutes, and there'll be a short period of time for questions direct to the speakers. At the end of the double session, we've got a big block of time to take questions, discussions, to hear your reactions, to have them discussed by the panel. But for anything that can't be aired in that way, please either use the hashtag or the email address in order to let us know what you think. We probably won't be able to respond, but we will aggregate the themes, and we will communicate them with other stakeholders and with the agencies that would potentially be involved if this were to go forward. I think that's it. That's all of my notes, and we hope that you will enjoy exploring this incredibly interesting and complex issue with us. Thank you.
Inviting the rest of our panelists, if you guys want to sit down, if it's easier to see the slides during the data session, that'll be fine, and we'll come back up at the panel where you can stay here. So thank you, everyone. Again, I'm not Elisabeth Bender, but we've worked together for over 2 decades, and she sends her regards. My first task is to talk about the earliest stage of embedding a biological diversity, everything from in utero to early epigenetic work. And what do we mean by that? So, exposure to early adversity likely changes, my disclosure slide, I guess the final one that had my disclosure slide was in there, so I'll just mention it. My disclosures are I have external support from Brainsway and from Takehome, and I've been on the Janssen and Verily advisory boards, and none of that relates to what I'm doing today. Exposure to early adversity changes likelihood of well-being through many different ways, and I'm going to talk about the epigenetics, and we'll hear more about other biology and neuroimaging components as we talk. Altered stress hormones and the cortisol regulation component is probably what the best is known about, and that's the story I'm going to tell today, FKBP5 and its regulation of the hypothalamic pituitary adrenal axis. This work's been shown by many people, including Linda Carpenter, Audrey Shrika, Christine Hine, Charlie Nemeroff's groups, and others. We also know it alters cardiovascular function, it alters brain connectivity, brain function, and brain structure. When we think about epigenetics, how can we think about this broadly? Here's a National Geographic cover of 2 identical twins, and though they're identical in many ways, we all know identical twins that have different personality traits, different interaction styles, and even different components of how they look, from freckles to other components. We know that epigenetic mechanisms now influence behavior, emotion, stress response, and the risk for psychiatric disorders. At the mechanistic study level in animal model systems, we can learn a lot more about this. Here's a famous example of the agouti mouse, in which basically the mother's diet related to antioxidants completely alters the agouti color of the offspring, and that's now known very mechanistically to be due to how CpG methylation occurs in the offspring's genome. When we talk about epigenetics broadly, we talk about 3 classes. One is non-coding RNAs, which I'm not going to talk about. Two is histone modifications, which broadly refer to the histone proteins that lead to wrapping and unwrapping of the chromatin, and I'm not going to really talk about that today either. And the third is DNA methylation, which would be the target of our discussion. Broadly, we're talking about the glucocorticoid receptor system, the hypothalamic pituitary adrenal axis. This was made most famous probably in the trauma world by Rachel Yehuda and some of the great work from that group, again with Christine Heim and many others, on dysregulation of the hypothalamic pituitary adrenal axis over time as a function of developmental adversity and lifetime adversity. And of the HPA axis, just to remind you, we talk about the hypothalamic pituitary component, which then releases ACTH, causing the adrenal glands to release cortisol, and glucocorticoids then feed back into hippocampus, amygdala, BNST, leading to the feedback of the system. And as with all systems, and really the world experts on homeostasis and allostasis, Bruce McEwen will talk in a little bit about feedback systems. We know early childhood trauma dysregulates these feedback systems and prevents homeostasis from acting in a normal way. At the cellular level, glucocorticoids cross the membrane, bind glucocorticoid receptors, which then act on stress-related genes. And that then leads to long-lasting effect on cellular response. Before I get into FKBP5 biology, here's a brief primer on how we think that epigenetics alters glucocorticoid receptor binding to the DNA and alters stress response. The idea here is that in a relatively region of the genome that is inactive, one has more methylation sites, and what DNA methylation at cytosine guanine dinucleotides does, it basically prevents proteins from binding to their normal sites. Also, more histone wrapping, or tighter histones, prevents those regions of DNA from being open. Specifically with stress, you get more cortisol, more activation of glucocorticoid response elements, which then lead to opening of these processes and changing an epigenetic state that lasts long term. What specifically I mean is by having more unmethylated CpG sites, proteins are now able to bind in ways they couldn't in the methylation conformation, and opening of histones allows regions of DNA to open. So when now cortisol is bound to glucocorticoid receptors, the glucocorticoid receptor, which is effectively a transcription factor, leads to increased stress-related gene transcription. And that can all be modulated by polymorphisms in the genes that alter regions of binding where the histones lead to wrapping or where glucocorticoid response elements are active. And the take-home message is we think that genetics that alters glucocorticoid response binding interacts with epigenetics together to essentially take the biological, the heritable risk combined with the environmental experience and alters the stress response system. FKBP5 is, at this point, kind of probably the best understood and sort of quintessential stress response gene that interacts with the stress system. Normally, again, at a slightly more detailed level, glucocorticoid receptors live in the cytoplasm and related to membranes and are prevented from being active in part by FKBP5, which acts as an inhibitor of GR translocation to the nucleus to activate stress activity. When sufficient cortisol comes into the cell, FKBP5 is dislocated from this complex. FKBP4 allows glucocorticoid receptors bound to cortisol to cross into the nucleus where they can now perform their function as a transcription factor. And then GR bound to cortisol acts stress-related genes. Well, it turns out one of the most stress-sensitive glucocorticoid receptor-sensitive genes is FKBP5 gene itself, such that more FKBP5 is produced, and you essentially produce more breaks within the cell to then inhibit the system. So at the same time, the cortisol system has a whole body feedback system. Every cell has this negative feedback system, and FKBP5 is at the center of that. And the take-home message is FKBP5 regulation by cortisol, by both polymorphisms at the gene level and through epigenetics, alters differential feedback on FKBP5 stress sensitivity. FKBP5 does not just regulate itself and regulate classic stress response genes, however. It's at the center of a whole host of many different protein complexes that are involved in everything from synaptic plasticity, neuronal function, synaptic transmission, inflammatory function, and many of the different pathways that we're going to talk about as dysregulated with early life stress. Furthermore, mechanistic studies and correlational studies in both humans and animals have shown FKBP5's function across these. Everything from neuroimaging and behavior related to threat as a function of FKBP5 genotype and epigenetics, more intrusive thoughts, impaired extinction of fear, and increased stress sensitivity and anxiety. And Jakob Hartmann, who did his PhD work with Matthias Schmidt at the Max Planck, where Elisabeth is, as well as his postdoc with our work at McLean, showed mechanistically that FKBP5 leads to increased stress and anxiety responses. Using genetically altered viruses, he overexpressed the FKBP5 gene in the amygdala and showed that those mice had more anxiety on an open elevated plus maze, more anxiety-like behaviors on a dark light box, and a number of other fear and stress-related behaviors. So how is the FKBP5 pathway tuned genetically and epigenetically? The take-home message is that genetic variants in FKBP5 alter the negative feedback of stress hormones at both the level of FKBP5 release and, we think secondary to that, cortisol feedback, such that one version of the FKBP5 gene, which I will simplistically call the risk allele, is associated with more FKBP5 responsiveness, i.e. there's less genetic methylation, more stress responsiveness, more FKBP5 RNA and protein made, more glucocorticoid response inhibition, and thus more GR resistance. So with that risk allele, more FKBP5 is made with stress in both cell systems and human and animal systems, and that increased FKBP5 is associated with more cortisol that then isn't turned off, isn't turned back down with further stress. So one can think that more FKBP5 induction is associated with more glucocorticoid resistance and less negative feedback. And Torsten Klingel, an assistant professor at McLean as well as at Göttingen University, has shown how this works at an epigenetic level. And the take-home message here is they showed in 3 different populations work from Torsten and Elisabeth and colleagues, Tanya Ivanovic and others at McLean at Emory University, as well as cohorts with Emory at Conti, the Emory Conti Center, as well as Rachel Yehuda's cohorts with Holocaust victims, demonstrated that increased epigenetic activity, i.e., sorry, let me say that again, an interaction of genetics of the FKBP5 gene and decreased methylation together were highly associated with increased risk. So the way to think about this is childhood trauma questionnaire in this cohort. So the more childhood trauma, the less methylation. The less methylation, the more FKBP5, the more glucocorticoid resistance, and we saw a very strong negative correlation in the risk allele genotype carriers. And this same pattern held up across these 3 different childhood trauma cohorts. What does this look like at the level of the phenotype? Again, we see across all the studies and much of what we'll talk about today is that increased level of childhood trauma is associated with increased PTSD symptoms, depression symptoms, and otherwise. But if you now stratify the increased risk of PTSD as a function of the FKBP5 genotype, those with the highest risk allele, those that are more likely to have decreased FKBP5 methylation, increased FKBP5 expression, increased glucocorticoid resistance, those also have much, much more sensitivity to childhood trauma compared to the group that has glucocorticoid sensitivity and a non-risk allele with relatively insensitivity. And we see no effect, interestingly, with adult trauma. And this is now, though this has not yet survived a whole genome association study, notably there haven't been essentially any large-scale genome-wide by epigenome or gene-by-environment genome-wide association studies. Over 25,000 samples and meta-analyses have now consistently shown this effect of FKBP5 interaction with childhood adversity on a number of stress-related outcomes. Certainly large-scale studies on depression and PTSD, but multiple other types of outcomes as well. Finally, Torsten Klingel, working with Mar Sanchez, has begun to show the similar effects in monkey models. And Mar at Yerkes has a fantastic model of intergenerational risk in which maltreated mothers have been shown naturalistically in monkeys to then have a much higher risk of maltreating their own offspring. And to unfortunately give this short shrift for a time, the take-home message is monkeys in which, even though they've had a history of maltreatment, are cross-fostered at the day of birth to a healthy, quote-unquote, nurturing mom, the monkeys remain differentially active and go on to show increased risk of maltreating their own, even though they only had the in utero exposure. And in addition to the behavioral outcomes of this cross-fostered cohort, they also show decreased FKBP5 methylation in the monkey homologous region of FKBP5 that lasts for up to 2 years in monkeys, which would be the equivalent of about 6 to 8 years in humans. So the early life stress in non-human primates also has been shown to shape the epigenetic profile in a way that's consistent with humans, and we have mouse data as well. So in summary, this model suggests that FKBP5 genotype interacts with developmental adversity such that those with the risk allele are more likely to have decreased methylation and more likely to have increased FKBP5 gene expression that is associated at a basic level with increased cellular responsiveness of a whole host of stress-related genes, changing neuronal function, changing brain function, and that we think then is associated with a whole host of effects of dysregulated homeostasis of the stress response system following developmental adversity. What would that mean for intervention? Well, at least at this, obviously we'll talk the rest of the morning about lots of ways we could think about intervention, but related to this specific model, if one knew how to dysregulate or disrupt, the overactive FKBP5 system through epigenetics or otherwise. And one example that Max Planck is working on is small molecule FKBP5 inhibitors that inhibit the FKBP5 glucocorticoid receptor interaction. And that's been shown in animal model systems to very robustly normalize the stress response pathway. So that, in summary, early exposure to early adversity leads to changes the likelihood of well-being, particularly in those with heritable risks. And FKBP5 has been one of the stories that is so far most mechanistically held up to understanding how this may work at an epigenetic and genetic and stress response pathway. Thanks so much to Elisabeth for all of her work over the last 2 decades, for giving me the honor to present some of our work together, and for all of the colleagues that have done this. Thank you so much. So given the time and that no one's rushing up to a microphone, I'm going to go ahead and move on to our next speaker, remembering that we'll have time at the end of this session and the end of the panel for more discussions. It's my great pleasure to introduce Ron Kessler to take us next into this story. Dr. Kessler really needs no introduction, but I'll say a couple words. He was trained in sociology at New York University, spent many years in sociology and epidemiology, and is now the McNeil Family Professor of Healthcare Policy at Harvard Medical School. He's published well over 1,000 manuscripts, is really the best-known epidemiologist in our field, is a member of the National Academy of Medicine, National Academy of Sciences, and recently elected to the American Academy of Arts and Sciences. Thank you, Ron, for all of your being a luminary in our field and helping guide the way, and particularly for being here today. Thank you, Ron.
Okay, thanks very much. It's nice to be here. So he told me to press the button and it would work. There it goes, it works. Those are my disclosures. I'm going to tell you a little bit about work we've done over the last number of years in epidemiologic studies that touch on the effects of childhood adversity. As some of you know, I'm the director of the World Mental Health Survey Initiative for WHO, where we do these big community epidemiological studies in countries around the world, mostly to consult with governments to tell them about the magnitude of the problem of unmet treatment of mental disorders in their countries. And we, as a result of this being coordinated, we pool the data across countries. We get very large samples. And childhood adversities is one of the things we've looked at from the beginning, the effects of childhood adversities on mental disorders in these cross-sectional surveys. And here are the childhood adversities that we asked about in the World Mental Health Surveys: interpersonal loss, parental death, divorce, separation, maltreatment, parental maladjustment of various sorts, and other extreme kinds of childhood adversities. These are not all the childhood adversities that exist. We don't have sexual trafficking or being in foster care or a number of other things, but these are the sort of the garden variety, relatively common childhood diversities. When you do a factor analysis of these things, you find that some of them hang together, which we've referred to as maladaptive family functioning childhood diversities: physical, sexual abuse, neglect, mental illness of parents, substance abuse, criminality, violence. These things are clustered very strongly, and it's hard to tease apart their individual effects because they're so highly correlated. Just to give you a rough feel, this is in, I think, 28 different countries around the world, nationally representative samples. If you say, how many people had any of those, this is the maladaptive family functioning childhood diversities. In high-income countries, 38% of adults say they had at least one of these in their childhood. In upper middle-income countries, 39%. Lower income, 39%. I mean, it's crazily systematic, and these are, as I said, 28 different countries surveyed over a decade. But for those who had any of them, the number of people who had multiples was 41%, 40%, 30%. And it turns out that when you look at the associations of childhood diversities with bad outcomes, it's those multiples that are important. Most people, it turns out, can do pretty well when one terrible thing happens to you, but the system gets overwhelmed when you have more than one, and depending how resilient the system is, whether you can have 2 or 3 or 4, is when the crack occurs. The broad overview of the World Mental Health results in lots of different papers looking at lots of different things are very briefly this. The maladaptive family functioning childhood diversities are associated in the cross-section with a wide range of retrospectively reported lifetime anxiety and mood disorders. Other childhood diversities, for the most part, are not. For those of us who are parents, I'm sad to tell you that if you die, it doesn't really have that much of an effect on your kid if that's the only bad thing that happened. If you commit suicide, it's a different matter. But if there's one other parent around, then there's no sort of violence and stuff, where most people can deal with one bad thing. Other childhood diversities, for the most part, not significant. It's difficult to tease apart the effects of individual maladaptive family function childhood diversity, as I said, because they are so highly clustered. However, the best fitting model, when you use these really large samples, and you'll recall back there, I think there were 60,000 people who we asked about childhood diversity. It's enormous numbers of people. The best fitting model is one that includes information about both type of adversity and number of adversities. In other words, it's which bad thing happens to you makes a difference. Sexual abuse is worse than neglect. But it's also they combine in a non-additive way. It's just that they don't add up. It's sort of this multiplicative, complex multiplicative effect. There's this cumulative exposure effect, which is sub-additive. And by sub-additive, I mean that the more bad things that happen to you, the worse it is. However, it gets less worse over time. It increases as a decreasing rate. Once you have 5 terrible things that happen to you, having a 6th doesn't really make things that much worse. You're already in bad shape. Now that has an interesting and important policy implication for intervention. And it's this, that if you go after people who are highly affected by lots of adversities, any one intervention is going to have no effect. Because whether you knock out the 7th adversity or the 6th and they still have 5, you might as well not bother. You've got to go after the whole ball of wax or nothing. And that means that many interventions that exist, interventions for treating the mother's depression. Well, that's fine if mother's depression is the only problem. But if there's a bunch of other stuff, not going to really do very much. So the effects of interventions, inversely proportional to the number of other childhood adversities you have. The similar associations we found in our data, and others have found as well, exist in predicting early onset of anxiety and mood disorders. It's not just do you have them, but early onset. Although interestingly, among people who have a lot of childhood adversities, if you manage somehow not to develop a childhood, adolescent, or early adult anxiety or mood disorder, you're still twice as likely as other people who've made it through that period of your life to develop one in midlife. So there's this long-term effect. It gets smaller over time, but it's there forever. Childhood adversities also predict severity and persistence of these disorders. Anxiety and mood disorders are more persistent than among people who have childhood adversities than other people who develop anxiety and mood disorders. A range of lifetime physical disorders are predicted by childhood adversities. Early onset of them, persistence and severity of them. So there's these pervasive effects, and Kerry already mentioned this to you. Now, all that stuff is retrospective, and asked adults who are sometimes in their 50s and 60s about what's going on. And I would note here that we've also done some studies. Here's a US study of 10,000 adolescents aged 13 to 17. And we found exactly the same results. So retrospective recall bias is to some extent an issue, but you find the same thing. There's also ways in which you could do studies. Here's an interesting study we did as part of the World Mental Health Series with Eli Karam in Lebanon, where we asked the same questions of adults, but we also included adolescents in the study. And we have information about objective occurrences. So we know in Lebanon, during the 2 wars that were in the lifetimes of the relatively young people in our sample, where you lived and which villages were attacked, which ones they had to evacuate, which ones not, and so forth. And you find a strong and consistent association between those objective experiences and anxiety and mood disorders. But interestingly, even in the face of that kind of population adversity, parental neglect and abuse was much more important than war. The observational. Here's an interesting study, which I think is a model for things we can do in the future, and I'll get to this in a minute. And it's a study of the Casey Family Foundation foster care program. It's the first model foster care program in America where anybody who gets into the foster care program, if you can get out of high school and get into college, Casey will pay for free for your college. They pay for medical school, for law school. You want piano lessons, they want piano lessons. I see your teeth are a little crooked. How about we'll get you braces? But it sort of gets you into a different kind of life rather than the people who are in foster care with 5 kids in a room living in the back of some working class person's home. We did this study 20 to 30 years after people were in foster care. We went back to the foster care agencies, looked at the records, and found Casey at the time only had a certain number of caseworkers. They have specially trained families who they pay who do this. And they only take kids where they have an opening. So on Tuesday, a kid came in who had a certain profile, and they had an opening, and they get into the Casey program. The next day on Wednesday, another kid comes in and looks just like them, no opening. They go to the state. So we essentially went through and carefully did essentially a waiting list control kind of thing where we matched people who got into Casey by the luck of the draw and those who didn't. And then we followed them 20 to 30 years later, and there are extraordinary effects. I mean, unbelievable effects. Effects that are so big that when we did a cost-effectiveness analysis, it's very clear that the extra expense of the Casey program more than pays for itself, in the sense that the reduced criminal justice expense to society of the people who were in Casey not being criminals, the increased income tax that are paid by these people because they have jobs, they make more money, the less child welfare, I mean, all kinds of stuff. It's clear that it's just a smart business move in the long run to invest in these kind of programs. Why don't we do it? Well, we don't do it is because it's a smart business move for the governor 20 years from now. The governor today wants to do something that's a smart business move for him today. And so you can't get a return on your investment right now. It's a long-term return on investment, and we sadly still live in a country where we're thinking of short-term wins. But it's clear that these are things you can do. And I'll come back in a little while if I have a second to that. That's still not really an experimental study. It's quasi-experimental, but I think as a practical matter, it's the best we can typically do. In rare cases, though, we get to do other things. And here's an example of one. The Moving to Opportunity study. Let me see, yeah. So it's the Moving to Opportunity study. This was a $70 million experiment carried out by HUD, where we went to 10,000 low-income, single-parent families in inner-city housing. Cabrini Green in Chicago, one of the famous ones, was in our study. And we randomized people to get a voucher to get a house in the suburbs for free. And then followed them for 15 years. again, it had an enormous effect on the kids, but it also is cautionary. Susan, we talked about this last night about sort of doing good is a lot harder than you think it is. This program had a great effect on the little girls. When they moved out of the inner city into other neighborhood, they were embraced as you poor little thing and sort of thing. But the boys, they were considered the official juvenile delinquent in the school, and they were extruded. So it turns out it actually had a bad effect on the mental health of the boys. They would have been better staying in the old neighborhood. So for all the, I mean, it's tricky kind of stuff. But experimental evidence that, again, when you manipulate environments in this way, you can have a big effect. Now, other kinds of studies don't have a chance to manipulate things, but they can still get something about causality indirectly by looking prospectively. And there are a lot of studies like this. Charlie, I think, is going to tell us about studies where you start with treatment samples, you ask people retrospectively about childhood adversity, and you can see going forward to predict stuff. This is one of our Army studies where we did the same thing. People's reports of childhood adversities at the time of coming into the Army are strong predictors of a whole bunch of mental health outcomes when they're exposed to stress. This is an interesting example of it because, as I said, childhood adversities combine in complex ways, and in this Army study, because we had an enormous sample, we had 35,000 new soldiers we interviewed, and we can take these dozen measures of childhood adversities and put them together in complex ways by essentially using a data-driven approach, using machine learning, artificial intelligence kind of stuff to do maximal combinations. And when you do that, and you come up with composite risk scores based on childhood adversity, what you see here is the x-axis is the 5% of people that we predict to be at highest risk, the next 5, the next 5. The 5% who we say are at highest risk, based on the multivariate, complicated combination of childhood adversities, they account for 35% of the suicide attempts over the next 2 years. I mean, it's an enormous, enormous concentration of risk. Is it causal? We don't know, but but certainly it's predictive. Now, we do know that there are causal effects. The kind of studies that Kerry mentioned, the animal studies where you manipulate, or the foster care study where we literally, we manipulate things, or the housing study. Are they biological? Is another matter. And again, Kerry told you about a lot of animal studies. There are now a number of human studies that are trying to do the same kind of things, observation. The UK Biobank, the Million Veterans Study, All of Us, ABCD, the Aurora Study, are all these big studies that start with some high-risk sample, collect a lot of biology before the onset of things you're interested in, and try to see, can you find some effect of the biological factors. The optimal design for doing these kind of things varies depending on what the question is. So if the focus is on determining the effects of your early childhood interventions, should we do the foster care program, or should we intervene in the schools, or do preschool things, or do things for new mothers that we do parenting skills things, then some kind of a fallback design would be ideal, like the Casey foster care study. And that design would be relatively easy to replicate. Just as an example, there are now in America 50 different model foster care agencies in lots of states. The 1% of kids in America are in foster care right now, $28 billion industry per year. To be able to go and 10 years back and find 20 foster agencies that took kids and didn't take other kids and follow those people up 10 years later would not be a difficult thing to do. And getting the biology post hoc is something that would be easy to do. You can use that as an instrumental variable to manipulate it. It's sort of a study waiting there to be done that won't take you 20 years to do, which a lot of long-term studies do if you actually do it the right way. And because model foster care agencies are modeled in different ways, some of them do have the piano lessons and some don't, and so forth, it's even possible to tease out what are the components that seem to be the critical ones that are important. If the focus is on using information about childhood adversities as a marker for adult interventions, then the perspective design makes more sense. And again, this is sort of what I get the impression Charlie and a lot of clinical people do. They start with childhood adversities. They say this is a group of people who are going to respond differently to treatment. We don't know whether that's really childhood adversity or it's somebody's recollection of childhood adversity, but in a way, it doesn't make any difference what it is. The people who tell us, for one reason reason or another, that they had terrible things in childhood, or ones that don't do as well, we can do something about that. In the case of a study like Aurora, that Kerry are involved in, where we have an extraordinarily rich and varied assessment of biological variables, as well as an enriched sample of people exposed to trauma. So we have 5,000 people who come into emergency departments after physical assaults or sexual assaults or motor vehicle accidents. And we put a watch on them and give them an app, and Kerry does this fMRI with them and stuff like that. We get blood samples. We have the opportunity to do that kind of stuff where you could sort of can fine-grain sorting out the biology from the environment. What is this? I can't read my own writing. Let's see here. It says something. Well, forget that. That must not be important. But those studies are cross-sectional, which is a problem for understanding something about causality. And one of the things that the Aurora study has the interesting potential to do, that's related to this thing I'm showing here about our World Mental Health Study, is following trauma survivors with all the biology that we have from them into second traumas over the next decade. It turns out that in the population, in the US population, at a point in time, 80% of the people who develop PTSD this year have had PTSD before. And one of the strongest predictors is trauma load in the past and having responded poorly. Not terribly surprising that the kind of people who have a tough time dealing with one trauma are going to have a tough time dealing with another trauma, but actuarially, that's sort of what's going on, and most of the people we study are recurrences, not first onset. So the ability to take this enormous amount of investment we've put in a study like where was it, 35 million now or something like that, to, instead of saying, well, the study's over, having that as a baseline for going forward has a lot of potential. Before ending, I want to say that everything I've talked about so far has to do with just sort of simple prediction. It doesn't have to do with what we're really interested in, and that is who's going to respond differentially to treatment. And as you know, there's this whole new world of precision medicine that looks at what are the characteristics, not just that predict bad outcomes, but that predict differential response to interventions for those bad outcomes. The statistical technology exists for doing that, and that's what these two citations are. However, it's very clear that you need enormous samples. So these little, like the Embark sample, 350 people with, it's hopeless. You just can't do that. You need at least 500, maybe 1,000 people in each intervention to be able to see something serious. And that typically would require an observational design. So for example, if we had that foster care study where you could go back inexpensively, get a couple thousand people who did and did not get treatment, you could do this kind of stuff. An example here is a prospective study we're doing of 3,000 veterans seeking outpatient treatment for major depression to predict remission. They have these different kinds of treatment. They get medication or psychopharm or some combination, and depending on which clinic they go to, they offer different things. We're doing a 45-minute assessment of them, and in using comparative effectiveness research, machine learning kind of things, we're developing what seem to be pretty darn powerful models for saying, instead of right now 30% of them do well on the first course of treatment, we could probably make it 50%. Just by doing the assessment and say, nope, that one gets the drug, that one gets the psychotherapy, this one gets both, in the first course of treatment. I mean, an enormous difference in observational studies. That's doable right now for not an enormous amount of money. We're doing a similar thing with 175,000 patients to predict suicide-related behaviors. We're interested in, among people who come into who make a suicide attempt, do they end up killing themselves? And what should you do? Should you hospitalize them? Should you put them in an intensive case management program? And again, in the VA, because they have 12 million patients, we have enough people that we can start looking at the kind of things that predict this. It's also possible sometimes to do controlled studies. So when you have these observational things, you'd like to be able to do a trial, but you need, as I said, immense trials. So here's an example. The Appalachian Mind Health Study, something I'm involved in, working in rural West Virginia, among people who are treated for depression, all in primary care. There's no psychotherapists out there in these little hill towns. So they all get cyclopharm. But it's the highest opioid death rate in the country, enormous comorbidity, and we know that combined psychotherapy and pharmacotherapy works better there. Who are the kind of people that's going to work best for? So we're essentially randomizing 5,000 patients. 2,500 are getting remote CBT on top of their cyclopharm, 2,500 are getting nothing, and we're following them for a year, developing precision treatment models to say who are the ones that should work for. There's never been trials of 5,000 people, you know? But we could do it for, it's a mere $15 million. And I say mere kind of not jokingly, because we have twice that much in Aurora, we're not going to get any place closer. So they're interesting designs that combine epidemiology with clinical stuff in thoughtful ways, taking advantage of the new technologies we have available, both for biomarker assessment and for doing fancy models. Exciting stuff on the horizon. I can't wait to see what happens. I'll stop with that. Thank you.
Thank you, Ron. That was terrific. It's outstanding. Thank you so much. Okay, so what we've now covered the importance, the broad topic that we're challenged with today. How do we move forward with understanding childhood adversity as a biological variable? We've just looked at it from an epigenetic perspective. We've looked at it very broadly with hundreds of thousands of samples from a developmental and epidemiological perspective. And now we're going to dive deeply into mechanism with the gentleman who started the field. So it's my great honor to introduce Bruce McEwen, who obtained his PhD in cell biology from the Rockefeller University. He's a member of the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. He was the president of the Society for Neuroscience from 97 to 98. I could go on for hours introducing Bruce, but I think relative to this is the important thing that we all appreciate, that in 1968, Bruce's Nature paper on the selective retention of corticosterone in limbic structures in the rat is really the beginning of our whole field of understanding the role of adrenal steroids and steroid pathways and stress. So thank you so much. Please join me in welcoming Bruce McEwen.
Thank you very much. It's great to be here with my wonderful colleagues and have a chance to talk to you. I'm going to continue the discussion of epigenetics, but from the standpoint of how the brain and the body communicate with each other through a variety of agents, not just cortisol, but multiple agents produced by the body affecting the brain, and of course the brain affecting the production of those agents. And I'm going to introduce you to the concept of adaptive plasticity of the brain. Our brains are changing all the time, for better or sometimes for worse. And then I'm going to take it to the question of early life adversity and how that affects these various processes. So if we go, then I have nothing to disclose. The big picture is, of course, that the brain is the central organ of response to experiences, some of which we call stressful, depending on our own personal history. A broader concept than stress is the notion that the body produces mediators such as cortisol and the autonomic, immune, neuroendocrine, metabolic systems and the neurochemicals in the brain as the process of adapting, actively adapting to what happens to us. And that term was termed allostasis by Peter Sterling and Joseph Ayer some decades ago. The idea that that leads to adaptation, on the other hand, the overuse or dysregulation of this nonlinear network of mediators can actually cause problems, leading to what we call allostatic load or overload, which can, culminate in disease. So as the bottom of the slide says, what keeps us alive can also kill us. Now, allostatic overload is sort of the metaphor is two heavy weights on a seesaw the seesaw remains in balance. We can maintain homeostasis for a period of time, but the seesaw itself is wearing out, and sooner or later it collapses and we have serious problems. Epigenetics, you've already heard. I happen to have started my career with Vincent Alfrey and Alfred Mirsky, who were really the originators of the idea that histones, which modify regulate the folding and unfolding of chromatin and regulate gene expression, they really describe the first modifications of histones that lead to gene expression or repression of genes. And our current leader at Rockefeller is a good friend named David Alice. And as you've heard, there are multiple mechanisms involving transcription factors, non-coding RNAs, the process of RNA editing, the methylation of cytosine residues, and the modification of histones. And as a further example of the development of individuality, a study that shows a pair of identical twins looking at methylation profiles on the left who are only 3 or so years old, showing virtually identical patterns of cytosine CpG methylation, but on the right, older identical twins where there's considerable divergence. And the term that's been introduced for this is the notion of non-shared experiences, because obviously twins go through life experiencing the same different things at different times, and even though they have many similarities, nevertheless there are differences as well as that emerge. The brain has the capacity throughout life for adaptive plasticity, and this involves the increased or decreased branching of dendrites on the left, the turnover of at least a subset of excitatory synapses in the middle, and a certain amount of neurogenesis, which occurs throughout adult life, particularly in the dentate gyrus region of the hippocampal formation. The neurogenesis can be inhibited by certain severe stressors, but it also is stimulated by regular physical activity involving mediators from the rest of the body. I'll tell you in a moment. Getting back to the idea that it's not just cortisol that's affecting the brain, the entire neuroendocrine system, gonadal, thyroid, and hormones like prolactin and growth hormone actually are able to affect the brain. In fact, there's some evidence that the brain, particularly the hippocampus, produces its own growth hormone, and it certainly responds to prolactin and to the metabolic hormones on the lower right, insulin-like growth factor, insulin, ghrelin, and leptin are all pro-cognitive and neuroprotective agents, and the brain is actually capable of making leptin as well, some of it. So it's emerging a very complicated and interesting story. The brain and the body are not that much separated from each other, and they communicate all the time. And that's true in the upper right with the entry of macrophages into the brain under some circumstances, not just brain damage, the participation of inflammatory processes systemically and also in the brain in depressive illness, but of course also in neurodegenerative diseases as well. So we can't ignore, just simply think about the brain as this isolated organ. It's functioning all the time with the aid of factors systemically like IGF-1, which is required for exercise to stimulate neurogenesis in the dentate gyrus. The human brain under stress, we know a lot from animal models and from structural and functional imaging studies of the human brain. The hippocampus is a brain region for memory and mood regulation, also to regulate the stress response. and repeated stress impairs neurogenesis and also can cause dendrites to shrink, which can impair the function of the hippocampus. The amygdala, on the other hand, is involved in fear and anxiety and aggression, and it actually helps to turn on stress hormones and increases, for example, heart rate. And chronic stress based on animal models and some evidence in humans can actually cause parts of the amygdala to expand and become more active. Whereas the prefrontal cortex, which is very important for memory and the ability to also regulate the autonomic and neuroendocrine systems, dendrites shrink with chronic stress. So what happens is that the brain under chronic stress is more vigilant, more anxious, perhaps less able to self-regulate, somewhat impaired or perhaps altered in the kind of memories it processes. Now, in a dangerous situation, this actually may be an adaptive trait. And yet, when the danger passes, if the system does not recover in some fashion and gets stuck, then we have an anxiety disorder, a depressive disorder that needs some kind of external intervention. So the concept was previously, if you saw structure changes in the brain, it must be damage. No longer, we know there is adaptive plasticity, that the brain is a dynamic organ, and if it doesn't respond to a changing environment, then some difficulties will develop, behaviorally or otherwise. Well, I've sort of said the shrinkage or growth of dendrites shown here happens in the basolateral amygdala on the right, also in the orbital frontal cortex, which has to do with vigilance. On the left, the shrinkage in the medial prefrontal cortex, the hippocampus, and the medial part of the amygdala all have effects on social interactions, self-regulation, and mood, and so forth. We have to bear in mind that there are profound sex differences, very subtle sex differences in the brain. Our sex is programmed not only by hormones secreted in development, but also by genes on the X and the Y chromosomes, and by the mitochondrial DNA that we inherit from our mothers. It also turns out that the entire brain has receptors for sex hormones in both males and females, and they exert their effects both at the nuclear level, but also non-genomically via signaling pathways. And as the slide says, many of these receptors mediate non-genomic effects on the cytoskeleton, neurotransmitter release, mitochondrial calcium buffering, and have nuclear effects, particularly in the very important inhibitory interneurons. And so the generalization then about males and females is that men and women, males and females, do many of the same things equally well, but they use different strategies, different neural mechanisms, and there are functional and structural imaging studies which support the existence of these subtle differences in circuitry that mediate these behavioral responses, both animal experiments and studies in the human brain. Now we come to adverse early life experiences, which we've begun to hear a lot about, which all of you know about. And I'm also speaking here as a participating member of the National Scientific Council on the Developing Child based here in Boston and Harvard under the direction of Jack Shonkoff. Now, there are many consequences, both systemic and neural, and I thought I'd just very briefly mention some of the more neurally related consequences. Of course, and of course we're talking about the epigenetic effects of these early life experiences. One thing that we and others have seen from animal models is that early life adversity, a model that was developed by Tali Barum at the University of California, Irvine, where the mother rat or mouse has restricted bedding, and so she's very irregular in her treatment of her young. That this irregularity leads to a reduced translational profile of messenger RNA, in this case from the hippocampus, from some of our studies. That suggests that the brain is less responsive as an adult, has less range of responses as an adult to challenges throughout life. From the human standpoint, as I'm sure many of you know, we can talk about chaos in the home, which impairs the development of self-regulatory behavior, can alter cardiovascular reactivity, blood pressure, lead to obesity. Work of colleagues such as Gary Evans at Cornell on this topic. And then adverse childhood experiences, including not only abuse, but also neglect and the transgenerational effects of poverty. Family poverty affects the rate of human infant brain. These are some of the headlines of papers that many of you already know. Association of child poverty, brain development, and academic achievement, and the famous Hart and Risley studies on the verbal stimulation and development of vocabulary. Also, then the hippocampus pays a price for early life adversity, smaller in poverty, but smaller with low self-esteem. I'll say more about that in a minute, as well as the increased risk for PTSD. At the bottom, there's an association between income and the hippocampus. So it's very sensitive to factors that relate not only to poverty, but also to abuse and neglect. This study, going back to Clemens Kirschbaum, taking people in the Trier social stimulation test, public speaking challenge, showing that while most people showed after one session and the open circles an habituation of their cortisol response to repeated TSSTs, there was a subgroup that actually produced more cortisol and never appeared to habituate. These were people who appeared to have low self-esteem or locus of control or sense of agency. And Jens Prusner, one of the people who participated in that original study, while at McGill, did a study of the hippocampus, showing that people with low self-esteem, both young and old, had a smaller hippocampus. And of course, there's some evidence from Roger Pittman and others that low self-esteem and a smaller hippocampus in particular is a risk factor for PTSD. And actually another thing that's emerged is that we know that the development of PTSD actually may be increased by low levels of cortisol at the time of the trauma. That might actually be prevented by elevating cortisol either during or shortly after the traumatic experience. So here, cortisol may be a protective factor, and you got to produce enough of it at the right time. The amygdala tends to be larger and more active in depression and anxiety disorders, larger in children living with a depressed mother. Work from Sonia Lupien and colleagues in Montreal. And then the prefrontal cortex. Accelerated development, maturation of the prefrontal cortex, sort of premature aging to deal with difficult situations may also reduce its ability to do the kinds of proactive planning and things that we appreciate our own ability to be creative and to function in a complex world. So the obvious idea is that the life course is a one-way street. It's a trajectory. This wonderful review by Neil Halfon and colleagues pointing that there are opportunities either to increase, improve health development, or suppress it. Some of the points listed on the slide, I'm sure we can all think about others, but that this is a one-way street. There's no such thing as true reversal, but there are possibilities for compensation. But prevention, of course, through things like the nurse-family partnership has been very effective. But another program that has promise is the Strong African American Families study of Brody and colleagues at the University of Georgia, where an intervention, 7-week intervention, improving parent-adolescent child-parent interactions, working on improving self-esteem and fighting bullying, actually reduced the incidence of a pre-diabetic status in young adulthood, and also prevented, on the graph on the right, the decline in amygdala and hippocampal volume that occurred without this kind of intervention. So perhaps this is another window of opportunity for affecting both brain and body health, and something that we should think about as a dissemination of a program on a wider basis. And finally, you're going to be hearing from Martin Teicher after the break, talking about the human brain and the notion of while we can't reverse, there are possibilities for compensatory changes to correct some of these problems or ameliorate some of these problems. So with that, I obviously acknowledge a huge number of people, but particularly the National Scientific Council for the Developing Child, and before that, a number of MacArthur networks, which broadened my perspective from a basic neuroscientist to someone interested in the broader aspects of health and disease. Thank you very much.
Thank you so much, Dr. McEwen, for just a wonderful overview of this field. So at this point, we'll take a 15-minute break and come back, please, and we will have Dr. Teicher and imaging, Dr. Nemeroff and treatment differences of childhood trauma, and then hope you'll join us for the panel discussion. Thank you.